Hi, everyone. This week we're going to be talking a little bit about microbiology. And some of you might be thinking, oh, wait a minute, this is human biology. Why are we talking about microbiology? And it turns out that microbiology, the bacteria and viruses associated with our bodies, have a significant impact on human health and in both positive and negative ways. I think we're so used to thinking about bacteria as being bad things. But one of the things that we've learned in recent information about bacteria is there's a lot of really good things about bacteria, a lot more than we ever knew. We're getting more tools to be able to study bacteria in laboratories, and some of these functions and what we know about bacteria and what they do and where they are really being changed. Our ideas are changing a lot. So the other thing is that there's a lot of things that we can do to influence the growth of bacteria and viruses in our body and again impact our health and disease. There's a vocabulary word here. It says microbe, and it says a microbe applies to a microscopic organism. The more common term you would hear, or the more formal term, would be microorganism. So that would be a living organism too small to be seen with the naked eye. And so just because it's a big word, we tend to shorten it and say microbe. But these can be bacteria, viruses, even protists, which are kind of eukaryotic single-celled organisms. And one of those is the organism that causes malaria. So we're going to talk a little bit about that. Another vocabulary word that's really important when you talk about microorganisms is the term pathogen. And pathogen means a disease-causing agent. Bacteria, virus, protist, other things can also cause diseases like worms and things. So we're going to talk a little bit about bacteria, and then we're going to talk about viruses. And I think there's just a common misunderstanding about bacteria versus viruses. So remember when we started off the semester and we talked about cells, and we talked about eukaryotic and prokaryotic cells, or eukaryotes and prokaryotes. So we said eukaryotes have a true nucleus, and humans, most organisms that you're familiar with are eukaryotic organisms. When we're talking about prokaryotic organisms, for your purposes, you can just say bacteria. So the bacteria are the prokaryotes. They don't have a nucleus. They're the pre-nucleus organisms. So what we understand about evolution and how life arose, it appears that prokaryotic cells were the first cells on the planet, and then through evolution, they developed a nucleus and compartmentalization and multicellularism that allowed eukaryotes to become a very dominant life form on this planet. So prokaryotic cells, they're single cells. They do not have a nucleus. They don't have the membrane-bound organelles like mitochondria, lysozymes, things like that are present in eukaryotic cells. There's actually, when you look at bacterial cells, there's three major shapes, and this shows you some pictures right here. In fact, you might not know, but I'm a Jeopardy fan. I love Jeopardy. And just the other day, there was a question about the three shapes of bacteria. So when you're looking under a microscope, you would see bacteria are usually round, which we call a coccus shape. They're elongated, which we call bacillus, or curvy, that we call spirals. And or you might hear the word spirillum. And so when you're looking at under the microscope, bacteria are usually pretty easy to pick out because of that coccus, bacillus, or spirillum shape. The other things to know about bacteria is they have a plasma membrane just like every other cell. They have a membrane on the outside that controls what goes in and out of the cell. Here they're showing you this. Oh, here we go. There's the plasma membrane. And inside of the cell is a cytoplasm. And just like all living organisms, they have to have nucleic acid. Now, you remember in eukaryotic cells, the DNA was divided up into, in humans at least, into 46 pieces called chromosomes. Well, in the bacterial cell, there's no nucleus to contain the chromosomes. In fact, bacterial cells are much smaller than eukaryotic cells, and they really have just one single circular chromosome. And that chromosome is kind of all folded up inside of the cytoplasm, and it's called the nucleoid. It's the nuclear area. But if you took that chromosome, that piece of DNA, you could shake it out and see one single circular chromosome. There actually is another piece of DNA inside of many bacterial cells called a plasmid. And as you can see, this plasmid is also a circular piece of DNA, but it's much smaller than the single circular chromosome in bacterial cells. And that plasmid might contain genes to give that bacteria superpowers. And one of those superpowers that bacteria can get is the ability to resist antibiotics, resist being killed by antibiotics. And so sometimes when bacteria have plasmids, it can be a really advantage to their reproduction. It can make them more fit. Let's see, another important bacterial structure is the cell wall. So here's the cytoplasm, the plasma membrane, just like eukaryotic cells have, but then most bacteria also have a cell wall. And the cell wall is a more rigid structure, and it helps support the bacterial cell. And one of the important things about the bacterial cell wall is those bacteria that have it, it's essential for life. And it turns out that some antibiotics, particularly penicillin and its relatives, attack the bacterial cell wall. And that's kind of interesting because when you're making a drug that you're putting inside of a patient, like a human, you want the drug, like penicillin, to kill the bacteria, but not kill the host, not kill the person that's getting that drug. And it turns out bacteria have this cell wall, and penicillin attacks the cell wall, but humans don't have a cell wall. So penicillin would not kill our cells unless you're allergic to it or something, but most people are not. And so that makes the cell wall a really good target for killing bacteria inside of human cells or other organisms. Okay. There's a lot of other information about bacteria and their parts in this chapter, but I just, in the interest of trying to make a shorter point than to just a complete understanding of bacteria, I'm going to leave some of these out. If you take microbiology, you'll get all that information. The one last thing I want to say about the bacterial cell wall is it turns out there's two different types. And the two different types, I'm not going to go into details about what makes them different, but the cell wall determines if you have gram-positive or gram-negative cells. And why this is important is gram-positive cells, when we're looking at them under the microscope, we can do this stain called the gram stain. And the gram-positive cells will stain purple, and the gram-negative cells will stain red. And it just gives us a first step into identifying bacteria. It also can give you some indication of what antibiotics the bacteria might be resistant to or might be sensitive to. So again, if you take microbiology, you'll learn a lot more about that. But the gram stain will tell if the cells are gram-positive or gram-negative. It helps us with identification, and it helps us understand about what antibiotics it's sensitive to. And it has to do with the cell wall. There's a little bit about the plasmids. And here's a little section talking about how do bacteria divide. And here's a bacterial cell in the middle of dividing, and we call that binary fission. Now, us as organisms, multicellular organisms like humans, we reproduce by sexual reproduction, and it takes maybe about 20 years for each generation to arise. But in bacteria, their next generation just occurs when one cell divides into two. So you can create a new generation of bacteria in a really short period of time. In fact, there's a nice little video right here that I recommend you're watching. Cell division in bacteria is called binary fission, and here's about a minute-long video that you can watch to understand a little bit more about binary fission. one of the things it talks about is how bacteria can divide in, even as short as 30 minutes in some cases. So new generations of bacteria can arise very quickly. And that becomes really important when we're talking about how do bacteria become antibiotic resistant. Well, if you reproduce very quickly, evolution also happens very quickly. And that's an idea that we'll be looking at a little bit more later in this lecture and in lab. Just a little bit about bacterial diseases. Just having bacteria growing in your body in the wrong place can cause different kinds of bacterial diseases. And so the disruption of your own tissues can be really an important thing that happens and causing signs of your disease. But a lot of bacteria also produce molecules called toxins. And these toxins are just waste products from the bacterial cells, and they just happen to be have negative effects on our bodies. So bacteria like Clostridium tetani that causes tetanus the disease is actually caused by a toxin that bacteria makes, and it causes our muscles to contract and not relax. And so it turns out that a lot of bacterial infections a lot of bacterial diseases are a result of bacterial toxins. That's the really short summary of bacteria. I just want to talk a little bit more about viruses. And so viruses are really different than bacteria. Here, the first thing it says is they're intracellular parasites, meaning they need a host cell to reproduce. Later, particularly in lab, I'm going to show you how we grow bacteria in some broth cultures or on agar medium, and you just give it the right food and the bacteria will grow. But viruses actually need a host cell. They need to be inside of a cell of some sort. And so, and that's because they're not made of cells themselves. They don't have a metabolism, and so they have to rely on the host cell. Think about children in a family that don't have jobs, they don't make any money, they can't buy a house, they don't know how to cook. They depend on you, their host. And so viruses do a lot of the same thing. They depend on their host cells. One of the big differences between the different kinds of cells, eukaryotes, prokaryotes, and viruses, is just their size. So here's a picture from your book that's showing you that eukaryotic cells versus the size of a prokaryotic cell, a bacterial cell, versus the size of a virus. And so viruses are much smaller. In fact, the microscopes we would use typically in a laboratory or a research lab or a teaching lab are just not powerful enough to magnify viruses. You need special microscopes. You need electron microscopes. So and that's actually the first thing we knew about viruses. We knew they caused diseases, but we couldn't find them under the microscope because they were so small. So it took a little longer to understand viruses than it took us to understand about bacteria. So viruses, they don't have cells, so they don't have plasma membranes, they don't have nucleus or anything like that, but they have an outer protein layer called a capsid. And so if we look at, this is an electron microscope of a virus, microscope picture of a virus, and magnified 60,000 times. We can see bacteria if we magnify it like 1,000 times. But here we've magnified the 60,000 times, and all these little dots are just separate proteins covering the surface of a virus. Here's a cartoon of the proteins that cover the surface of the virus, and collectively these proteins are called the capsid. Now, what's the other major structure of a virus is what's inside of the capsid is DNA or RNA. And so you can see they made a little hole there so you could see the inside. So viruses also have nucleic acids. Now remember, we said bacteria and humans all have DNA, and then we make RNA. But it turns out that viruses have DNA or RNA. And again, that's something that will be relevant if you took microbiology and you got into more detail about understanding that. Some of the other parts that viruses might have, they might have these structures called spikes. And so they're showing you here, there's some spikes, here's some spikes on the surface of this virus. But they all are going to have some DNA or RNA. They're all going to have a capsid, a protein coat. Some viruses have an outer layer called an envelope that's a membrane structure. And then in that envelope or in the capsid itself, they might have some of these other proteins sticking out that they call spikes. It turned out that the spikes in the coronavirus, the one that causes the COVID-19, made that virus kind of look like it was wearing a crown, and corona is another word for crown. So the coronaviruses got their names by the appearance of the virus under a microscope, that it had all these spikes. This diagram, even though I don't go into too much detail about it, it's just showing you that a virus has to infect a host cell. In this case, a virus is infecting a bacterial cell. So even bacteria have viral infections. And what it's showing you is that virus has to multiply inside of the host cell before popping out to infect more cells. So we say it's an obligate intracellular parasite. Has to be inside a host cell. Let's see, what else do we have? Yeah, and so down here we just have some good questions. I think 1 and 3 would be the ones for you to answer on this. I'm going to stop there and go to the next video.